At the molecular level, there is a circle of life. DNA makes RNA. RNA makes proteins that make more DNA. DNA stores the genetic information for making proteins. In longer form, the DNA code is transcribed onto RNA. The RNA carries the genetic code to ribosomes. The ribosomes use RNA to make proteins. The protein enzymes assemble more DNA and RNA. Protein enzymes also catalyze the formation of carbohydrates and fats that support the cell function that protects and nourishes the DNA in the cell nucleus. This is the circle of life at the molecular level. DNA, or deoxyribonucleic acid, is a chain of paired nucleotides that stores the code to build all the proteins that, in the end, build the entire organism. DNA is coiled up in the nucleus of every cell in its iconic double helix form. The nucleotides are bonded together in complementary pairs to preserve the coding sequence. If there is a coding mistake during DNA replication, a proofreading enzyme will detect the mistake when the pairs do not complement each other. The proofreading enzyme will replace the copying mistake with the correct nucleotide that complements the DNA template strand that is being copied. RNA, or ribonucleic acid, is a short single spiral of nucleotides that is transcribed from the DNA. There are many different types of RNA, but for this video we will focus on messenger RNA, mRNA, and transfer RNA, tRNA. The messenger RNA brings the DNA code for a protein to a ribosome. The ribosome translates the RNA code and assembles the protein per those instructions from amino acids that are brought to the ribosome by transfer RNA. The whole DNA sequence of an organism is called its genome. The genome of an organism is usually not one continuous DNA strand, but is distributed among multiple DNA strands called chromosomes. In sexually reproducing organisms, the chromosomes come in pairs, one paternal chromosome from its father and one maternal chromosome from its mother. The base informational unit of a genome is a gene. One gene contains the information to build one protein or several variations of one protein. An organism's genome size, its number of chromosomes, and the number of genes in its genome are not related. A human genome has 3.2 billion nucleotide pairs and 23 chromosome pairs. Some very primitive protozoans like the Dubai amoeba have huge genomes, over 600 billion nucleotide pairs. The huge genome of the Dubai amoeba is likely the result of the identical chromosomes that have been copied hundreds of times and never discarded. A higher animal would never survive such a mutation, but the primitive amoeba survives just fine with its DNA overload. Column 2 of the table gives the genome size of several species of animal, plant, bacteria, and viruses. The gene count of an organism tells us something about how many proteins it can make. At latest count, a human being has about 20,000 genes, so can make at least 20,000 proteins, and probably more given that, with alternative exon splicing or RNA editing, one gene can often produce several varieties of a protein that perform separate functions. Column 4 of the table gives the gene count of several species. Note that the mammal species all have around 20,000 genes that produce similar, if not identical, proteins. Plants must have more genes, as they must include photosynthesis and a more elaborate immune system than animals due to their immobility. Frogs and blowfish excepted. An animal can't make its own flesh poisonous to a predator as some plants can. The chromosome counts are random and species-specific to even closely related species. Humans have 23 pairs of chromosomes, hence the name of the popular genetic testing company 23andMe. Chimps, gorillas, and orangutans have 24 pairs of chromosomes. Shifts in the number of chromosomes prevent interspecies breeding. Cattle have 30 pairs of chromosomes, pigs have 19 pairs of chromosomes. Horses have 32 pairs of chromosomes, while donkeys have only 31 pairs of chromosomes. A mule, the offspring of a male donkey and a female horse, is viable but is sterile because of its chromosome mismatch. It is thought that chimps and humans had a common ancestor 6 to 10 million years ago. Sometime after 6 million years ago, and possibly only 1 to 2 million years ago, there was a shift in the number of chromosomes between the human line of descent and that of the great apes. The chimps retained the 24 chromosomes of the common ancestor. In the human lineage, a chromosome shift occurred. The human lineage was reduced to 23 pairs of chromosomes. Neanderthals and Denisovans are also believed to have 23 pairs of chromosomes. We may never know exactly when the human chromosome shift from 24 to 23 chromosomes occurred because DNA is not preserved for millions of years. What geneticists think happened is that 2 chromosomes of the common ancestor to chimps and humans fused. This fusion scenario is illustrated in the figure. In option A, the common ancestor to humans, chimps, and gorillas had 24 pairs or 48 total chromosomes. At some point after the human line diverged from the chimp line, the 2 chromosomes of the common ancestor fused to form the single human chromosome 2. An alternative scenario is shown as option B. In option B, the common ancestor to humans, chimps, and gorillas had 23 pairs or 46 total chromosomes. The human line retained the 46 total chromosomes. But after the lines split, chromosome 2 of the common ancestor split independently in the chimp and gorilla lines to give both chimps and gorillas 24 pairs or 48 total chromosomes. Option A, in which the common ancestor had 48 total chromosomes, is thought to be the more plausible scenario. Option A is less complicated. It allows for chimps and gorillas to retain the 48 total chromosomes of the common ancestor. Option A also explains why orangutans also have 48 total chromosomes, as orangutans are thought to have a common ancestor with humans, chimps, and gorillas 12 to 14 million years ago. The sequencing of the genomes of both humans and chimps reveal that chimps are the most closely related living species to humans. When we line up the 23 human chromosomes against the 24 chimp chromosomes, we note the only major difference between the 2 karyotypes is at human chromosome 2, the chimp has 2 chromosomes. We could name these 2 chimp chromosomes 2A and 2B or 2P and 2Q. The comparison of the karyotypes of humans and chimps is shown in the figure. Note that chromosome 2 is circled for a deeper examination. In the figure, we have an illustration of the human chromosome 2 put side by side with the 2 chimp chromosomes, labeled 2P and 2Q, that are thought to have fused together in humans since the time of the split from the common ancestor. The black and white banding on the chromosome indicates positions of genes and related DNA sequences. We can see the banding on human chromosome 2 lines up nearly band for band with the banding on the chimp chromosomes 2P and 2Q. A detailed study has shown that the suspected fusion patch on the human chromosome 2 has a residual telomere-like sequence of non-coding DNA that almost identically matches the telomere sequence at the ends of the chimp DNA. This telomere sequence matching lends further evidence that supports the option A scenario, the scenario that suggests that both humans and chimps descend from a common ancestor that had 24 pairs or 48 total chromosomes, and that at some unknown time since the split, the human line fused 2 of the ancestor's chromosomes into the single chromosome 2. In the figure is an illustration showing the chromosome shifts of the 3 great apes, orangutan, gorilla, and chimpanzee, compared to human chromosome 2. The illustration shows the similarities and some difference of all 3 great apes to the human chromosome, with the genetic bands on the chimp chromosome most closely lining up with the genetic bands on the human chromosome 2. A common ancestor between apes and humans millions of years ago does not rule out that God created evolution, or that He created Adam and Eve, the first humans in the Bible. Science and naturalistic explanations tell us how God did things, but they don't explain why God did them that way. Gibbons and siamangs, a whole family of lesser apes that live in Southeast Asia, are thought to share a common ancestor with the great apes between 16 and 20 million years ago. we might expect that gibbons would have 48 total chromosomes like the great apes. Unfortunately, multiple species of closely related gibbons have a massively mixed chromosome scheme. The ch016h gibbons from the forests of Burma and easternmost India have 38 total chromosomes, or 19 chromosome pairs. The nle gibbons from the forests of Laos and Vietnam have 52 total chromosomes, or 26 chromosome pairs. The hpi gibbons from Thailand and Cambodia have 44 total chromosomes, or 22 chromosome pairs. The ssy gibbons from Malaysia and Sumatra have 50 total chromosomes, or 25 chromosome pairs. The hmo gibbons from the forests of eastern Java have 44 total chromosomes, or 22 chromosome pairs. All this variation in chromosome pairs shows that, even in closely related higher mammals, chromosome shifting is common and not likely to produce major differences in body plan as long as the underlying genes and genome remain intact. Plants have random numbers of chromosomes as well. Crossbred or hybrid plant species often retain the full set of chromosomes from all the donor species. This is the case with common wheat or bread wheat, Triticum aestivum. Bread wheat is domesticated cereal crossbred during ancient times from wild einkorn wheat, Triticum boioticum, and emmer wheat, Triticum turgidum. Emmer wheat is a natural hybrid of a different species of wild einkorn, Triticum urartu, and an unidentified cousin of spelt wheat, Aegilops speltoides. Emmer wheat has 14 double chromosome pairs or has a tetraploid chromosome structure, 4 chromatids. As all 3 ancestor wheat varieties of bread wheat had 7 chromosome pairs each, the result of the crossbreeding is that bread wheat has 21 triple pairs of chromosomes, or has a hexaploid chromosome structure, 6 chromatids. Bread wheat should have all the genes and be able to produce all the proteins of all 3 of its ancestor wheats. However, this result is not the case, as many of the bread wheat genes have been suppressed, fragmented, or lost through thousands of years of domestication. 2 things are certain about bread wheat. First, bread wheat produces larger grains than any of its wild ancestors. And second, the grain seeds do not shatter or fall off their ears naturally as their wild ancestors do. Bread wheat ears must be manually or mechanically shattered to release their seed. This makes it easier for farmers to harvest bread wheat, but it makes bread wheat unable to grow in the wild. Protozoans are the most primitive form of eukaryote and are single-celled by definition. Protozoans can form colonies. Protozoans like Paramecium and amoeba species reproduce both sexually and asexually. Both Paramecium and amoeba are polyploids, meaning they have multiple copies of the same chromosome. As we mentioned, the Dubai amoeba has a genome that is 670 billion nucleotide base pairs long, giving it the longest genome of any known organism. A human genome is 3.2 billion base pairs. A lot of the amoeba's genome is hundreds of copies of the same chromosome. Despite its grossly large genome, the Dubai amoeba has only about 12,000 genes, so about half the gene count of a mammal. Common amoeba, Amoeba proteus, generally reproduce asexually by simple cell division, although reproduction by conjugation and transfer of nuclear material does happen on occasion. Paramecium also reproduce asexually for the most part. A single paramecium can divide asexually for up to 200 generations without exchanging genetic material. Asexual reproduction results in a population of clone paramecium. However, after 100 to 200 generations without the exchange of nuclear material, the population generated from the single paramecium will grow old and die off. Like amoeba, paramecium are polyploids with multiple copies and sometimes hundreds of copies of their chromosomes. Paramecium have a genome in the range of 72 million nucleotides and have around 39,000 genes, so they could potentially produce up to 50 or 60,000 proteins. Paramecium typically have 2 nuclei, a macronucleus and a micronucleus. The macronucleus is involved with the day-to-day transcription of DNA into RNA that run the cell functions. When it's time for paramecium to divide asexually, the macronucleus divides amitotically, meaning no mitosis. The genome in the macronucleus replicates itself without compacting into visible chromosomes. The nuclear material is divided equally, and the whole mess is pulled in two directions, and the whole macronucleus divides in two. Biologists believe the macronucleus serves as a backup copy of the genome for the micronucleus. The micronucleus is involved more with sexual reproduction. Before the micronucleus undergoes its meiotic division, proofreading enzymes check to be sure that the genome in the micronucleus is the same as in the macronucleus. Any differences are cut out and rejected as mutations or possible corruptions by viruses. The micronucleus then undergoes meiosis to form up to 8 haploid micronuclei. The haploid micronuclei can be exchanged with another conjugating paramecium partner. So there is a sexual exchange of DNA. The sexual partners break the conjugation once after each has received a haploid micronucleus from the opposite partner. The new haploid micronucleus moves to meet one of the haploid micronuclei that remains inside its mother. When two haploid micronuclei meet, they fuse and their nuclear material combines and recombines into a whole new genome. This new genome replicates and divides into a new macronucleus and micronucleus, and the older macronucleus and micronuclei dissolve. The paramecium begins a whole new cycle of asexual division with its new genome to produce a new healthy, multi-generational population of asexually reproducing paramecium. Viruses are not organisms. They are infective agents. They are free-floating strands of DNA or RNA wrapped in a protein coat or capsid. Outside of cells, viruses do not breathe, metabolize, grow, reproduce, or carry on any function that would be associated with being alive. Viruses often specialize to infect only one species or one type of cell. There are far more types of viruses that infect only bacteria than human or animal cells. When a virus finds its way into a living cell that it is specialized to infect, by trickery or other means, it inserts itself into the DNA of the host cell and hijacks the whole metabolic process of that cell into becoming a virus factory that makes millions of copies of that virus. The host puts on the protein coat for every replicated virus DNA or RNA strand and releases the new viruses to seek out and infect similar cells. This is the life cycle of the virus. Find a suitable cell, hijack its DNA, make more viruses that will seek out new cells to infect. Viruses carry only enough information to carry out these functions, so they can be very short. The shortest known virus, the porcine circovirus, a pig virus, has a single-strand genome only 1,726 nucleotides long. The porcine circovirus has only 2 genes and can make only 2 proteins, but this short strand of nucleotides is sufficient to hijack a pig cell and cause it to make millions of new porcine circovirus strands. The virus with the longest known genome is the Pandora virus. The Pandora virus specializes in infecting the protozoan amoeba. The Pandora virus genome has 2.4 million nucleotides and has up to 2,500 genes. For what it does, I have no idea why it needs to be so long. Neither does the Pandora virus. At least for a cell, viruses are like a horror movie come true. Invasion of the Body Snatchers or one of the zombie films. I think we have all experienced the common cold or one of the influenza strains and can understand how bad we feel when our cells are hijacked and turned into virus-making factories. As we saw with the Dubai amoeba, a lot of the genome is duplicate copies of DNA sequences. Some of these duplicates are copies of useful genes that code for proteins. These might be useful if one protein gene gets corrupted, a different copy might be used to make the protein. Some of the genome is repeating copies of the same letter at the chromosome ends, the telomeres. As we saw, These repeating sequences on the telomeres need to be there because the telomeres are shortened with each cell division. Some of the other non-protein coding DNA might be useful in RNA transcription regulation, to speed up or slow down the rate at which RNA is transcribed. A lot of the DNA sequence that does not code for proteins. Geneticists just don't understand what it does. For now, it is called junk DNA. We may find its real purpose sometime in the future. We may find the non-coding introns in DNA are just random nonsense. Genetics and DNA sequencing is such a new science that we don't know everything about genetics. The human genome has about 3.2 billion nucleotide base pairs. Less than 2% of the human genome codes for functional proteins. In a human being, or any mammal for that matter, the average gene coding sequence is just over 1,100 nucleotide base pairs long. If a human has 20,000 genes, this means that all the protein coding genes could be written on just 23 million nucleotide base pairs. What are the other 3.17 billion nucleotide pairs for? At this time, genetic scientists don't know. They call the extra, apparently non-coding DNA junk DNA. At the website Genetic Education, Dr. Tusher Chauhan posts the article Junk DNA Definition, Structure, Function, Percentage, and Examples. In that article, Dr. Chauhan provides a list of possible reasons for junk DNA, or as he calls it, non-coding DNA. Dr. Chauhan's list of possibilities include regulation of gene expression, genome regulation, maintaining genome integrity, role in the process of evolution, functional non-coding RNA, regulation of transcription and translation, origin of DNA replication, centromere and telomere regulation of structural chromosomal elements, scaffold attachment regions, regulatory sequences, provides biodiversity, prevent aging, protein and enzyme binding sites. The possibilities may explain some of the reasons for non-coding DNA. I am sure there may be other purposes for non-coding DNA that Dr. Chauhan left out. There is also the possibility that some of this non-coding DNA is junk. This junk DNA may be fragments of once functional DNA that is no longer needed, but was never removed from the genome. Some of this DNA junk may be the remains of virus DNA that was never purged from our genome. Some of this junk DNA is mutations that never had any function. There is the possibility that much of the non-protein coding sequence are random nonsense. Maybe they are not random, and we just have not figured out what they're for yet. If these non-protein coding sequences are truly random junk, it is an awful waste of genetic material. Just like the voids between galaxies is an awful waste of space.
Thank you for watching. If you enjoyed this video and want to help me make similar content, please like and subscribe.